Good morning. I am Dr. Amitha. Today I'll be taking the class on theories of mineralization. So, before we go on to the theories of mineralization, let us look at what all comes under the contents. You have to know about the different modes of bone formation, intramembranous ossification, endochondral ossification, then about the mineralization and the theories associated with the mineralization. Then we'll come to the inhibitors of mineralization. Coming to bone formation. Bone formation is carried out by the cell osteoblast. There are two different modes of ossification. One is intramembranous, the other one is intracartilaginous or endochondral bone formation. The direct formation of bone within highly vascular sheets of condensed primitive mesenchyme is called as intramembranous ossification. This occurs in flat bones of the skull and clavicles, and it begins towards the end of the second month of gestation. Now, what happens during the formation of bone matrix? At the site where the bone will develop, there is the presence of a loose mesenchyme consisting of widely separated stellate cells with interconnecting cytoplasmic processes. This develops in association with capillaries that grow into the mesenchyme. Mesenchymal cells in the center thus becomes round and basophilic with a thick interconnecting cytoplasmic processes. These mesenchymal cells then differentiate into osteoblasts and they secrete the organic bone matrix or the osteoid. Once surrounded by the bone matrix, these cells are then called as the osteocytes. The matrix soon begins to calcify. The earliest crystals appears in association with the extracellular matrix vesicles produced by the osteoblast. Crystal formation subsequently extends into the collagen fibrils in the surrounding matrix. Even after calcification, osteocytes obtains nutrition by diffusion along the canaliculum. The first formed bone is an irregular shaped spicule. This bone then gradually grows into long anastomosing structures called as the trabeculae. These trabeculae get extended in a radial pattern leading to the formation of spongy bone. These trabeculae encloses the local blood vessels, so such an early membrane bone formed is called as a woven bone. Now here, few of the mesenchymal cells remain undifferentiated. Before they disappear, they leave a layer of flat cells called as the osteogenic cells on the trabeculae. In highly vascular areas, whereas these osteogenic cells will give rise to osteoblast. But in areas with no capillary blood supply, they form chondroblast, which lays down the cartilage. So, in this way, new layers of bone matrix gets deposited on the pre-existing bone surface. Osteogenic cells from the spicule gets formed, and on the trabeculae, this process gets repeated again and again. In the vascular areas, osteoblast gets formed, bone matrix gets formed. Like that, new layers of bone matrix gets deposited on the pre-existing bone surface. In this manner, the process gets repeated. So, new bone gets deposited on one surface, whereas bone gets resorbed on the other surface, altogether resulting in the remodeling of the trabeculae. This remodeling of the bone helps to maintain the shape and the size of the bone throughout the life of the individual. Continuous oppositional growth and remodeling of the bone results in the cancellous bone getting converted to compact. Cancellous bone remains in the central portion of the bone as trabeculae does not increase in size. Vascular tissue in the cancellous bone gets converted to red marrow. So as layers of bone tissue is getting formed by this very process of apposition, the trabeculae starts getting thickened. The soft tissue spaces, they get narrowed. Narrow canals lined by osteogenic cells are getting formed, which encloses the blood vessels. In this manner, conversion systems gets formed. So this is a picture showing the primitive mesenchyme. Mesenchymal cells, blood supply rushes into this area, resulting in the mesenchymal cells getting differentiated into osteoblast. Cells gets trapped within the bone matrix due to the rapidity of the bone formation, and later on, the mesenchymal tissue gets converted to the bony tissue. Now let us look at the intracartilaginous or endochondral mode of bone formation. These are the various steps involved. There is the formation of cartilaginous model, formation of bone collar, followed by the formation of periosteal bud and the medullary cavity. This is followed by the formation of a secondary ossification center, followed by mineralization. So at the site where the bone is to be developing, under avascular conditions, the mesenchymal cells in this site will condense and get differentiated into chondroblast. The chondroblast will lay down the cartilage matrix. In this manner, a hyaline cartilage model gets formed. Now, this model is surrounded by the perichondrium, followed by the mineralization of the matrix. This matrix later on increases in length as in by interstitial growth by the repeated addition of chondrocytes along with the production of additional matrix by daughter cells. This results in an increase in thickness or a positional growth takes place due to the further addition of matrix to its periphery by new chondroblast derived from the chondrogenic layer of perichondrium. Now, in the midsection of the model, capillary starts growing into the perichondrium as the inner layers of perichondrium gets differentiated into osteoblast, bone collar gets formed. The perichondrium is now getting referred to as a periosteum. Vascularization at the midsection of the cartilage model happens. Chondroblast here will resorb the mineralized cartilage matrix. Periosteal capillaries and osteogenic cells will invade the mid-region of the model, resulting in the formation of a periosteal bud. There, the primary ossification center has developed. Osteogenic cells in the periosteal bud will get differentiated into osteoblast, which will result in the deposition of bone matrix on residual calcified matrix of cartilage, resulting in the formation of a network of mixed spicules or primary spongiosa. Osteoclast now breaks down the newly formed spongy bone, resulting in the formation of a medullary cavity. This results in the center of the shaft, that is in the diaphysis, resulting in the formation of a medullary cavity in this region, whereas the two ends of the developing bone gets composed of a cartilage, that is in the epiphyseal part of the bone. The formation of the bone here is the same as that occurs in the primary ossification center, but no medullary cavity formation occurs in the epiphysis. Hyaline cartilage at two places will be present, that is on the articular surface as articular cartilage, and at the junction of epiphysis and diaphysis as epiphyseal plate. So this is a picture showing the endochondral mode of ossification. This slide shows the different two modes of ossification. The intramembranous at the top portion and the bottom part of the picture showing the endochondral mode of ossification. Now coming to the mineralization, process of deposition of insoluble calcium salts in the tissue is called as mineralization. There are two main mechanisms involved in this very process of mineralization. They are the booster mechanism and the seeding mechanism. Booster mechanism states that a local increase in the level of calcium or phosphate causes supersaturation, resulting in the precipitation out of the solute from the tissue fluid to the site of mineralization. Seeding mechanism states that there are certain substances like collagen which acts as seed or nucleators to attract the calcium and phosphorus ions. So based on these two mechanisms, three theories have been proposed. Robinson's alkaline phosphatase theory, seeding or nucleation theory, matrix vesicle theory. Now, Robinson's alkaline phosphatase theory is based on the booster mechanism. Evidence for this theory is based on the observation that alkaline phosphatase was always found in areas of mineralization. Now, this enzyme hydrolyzes the organic phosphate containing substances, resulting in an increase in the level of local inorganic phosphate concentration, leading to supersaturation and precipitation of the phosphate ions from the tissue fluid. Alkaline phosphatase also helps to remove inhibitors of mineralization like pyrophosphate. The drawback of this theory is that this enzyme is also found in areas which do not mineralize. Coming to the seeding or nucleation theory, which is based on the seeding mechanism, the most important seed is collagen. Other seeds are phosphoproteins, glycosaminoglycans, chondroitin sulfate. Evidence for collagen as a seed comes from the fact that deposition of calcium and phosphate ions occurs first in relation to the cross-banding of collagen. Now, there are evidences against this theory. How does the collagen of the soft tissue do not initiate mineralization? And how does mineralization occur in the absence of collagen in the enamel tissue? So the explanation given for these two evidences against collagen was that in soft tissue collagens, the diameter between the collagen molecule is smaller than the hard tissue collagen, thus preventing the entry of phosphate ions and thus preventing mineralization of soft tissue. The explanation was given for the occurrence of mineralization in the absence of collagen in enamel tissue as in enamel, there are other seeds like phosphoprotein acting as seed. Now, coming to the last theory, that is the matrix vesicle theory, it states that amorphous calcium phosphate is formed within the secretory matrix vesicle of osteoblast. Alkaline phosphatase present within these cells serves to increase within the cell, which helps to increase the phosphate ion concentration within the cell. Mitochondria releases calcium from its stores, leading to the formation of amorphous calcium phosphate within the secretory vesicle. They are then released within the nucleating sites within the collagen fiber. So this theory incorporates both the boosting and the seeding mechanism. Now, coming to the inhibitors of mineralization, example is pyrophosphate. Pyrophosphatase helps to remove these inhibitors of mineralization or pyrophosphate from the nucleating sites to facilitate the very process of mineralization. So, in this way, the growth of crystals, size, shape, etc. gets regulated by inhibitors, which gets attached to the crystals to prevent their growth or alter their shape. So we have come to the end of this class on mineralization. Thank you.